Good afternoon, everyone. Thanks for being here and welcome to the final bi-faculty, for-faculty talk of the spring semesters. Now, we launched this series in 2016, and the idea was to bring together colleagues to share research that they're doing so we can learn from one another, and in the process, we can build community. And what I had heard over the last several years is that this is what people really are hungry for, is to know not just what people that they are in their same research groups or departments are doing, but what's going on at the university. How can I learn more? And how can I be part of this larger intellectual community? We built this series to create an avenue to do this, and it's worked remarkably well. So thank you all for joining. Now, it was about 5 years ago yesterday that Dr. Phyllis Dennery gave one of our very first bi-faculty, for-faculty talks. And I know that she's registered to be with us today. So thank you, Phyllis, for helping make this series such a success. Now, while many of you who registered for today's talk have attended past sessions of the series, I also noticed a number of new names, so welcome. Now, thank you all for joining what I know is going to be an incredibly important and interesting and insightful talk. I wanted to let you know that following the talk, there will be time for Q&A, and I'll ask you to use the raised hand function at the time. I'm not able to see all of you, but if you use the raised hand function, I'll be able to see you, call on you, and then we can engage in some Q&A. It's now my distinct pleasure to be able to introduce our featured speaker. Dr. Wafik El-Deiry is an internationally recognized expert in cancer biology who discovered the molecular mechanism that forces cells harboring genetic mutations to discontinue, an absolutely key process that limits the number of cancers a human will develop in their lifetime. He has further discovered how this mechanism is subverted in settings where human cancers do occur. His discoveries have led to the development of novel drugs that have entered more than 20 clinical trials. Wafik joined the Brown community in 2019, and today is Associate Dean for Oncological Sciences at the Alpert Medical School, Director of the Cancer Center at Brown University, and Director of the Joint Program and Cancer Biology at Brown University and its affiliated hospitals. He currently holds the Mencoff Family University Professor at Brown University. In addition, he's a licensed and practicing physician scientist and a medical oncologist with clinical privileges at the Rhode Island Hospital and the Miriam Hospital here in Providence. Prior to joining Brown in 2019, he served as a Deputy Cancer Center Director for Translational Research, Professor of Oncology, and the William Wikoff Smith Endowed Chair in Cancer Research at the Fox Chase Cancer Center in Philadelphia. He has more than 400 peer-reviewed publications and 5 edited books, and is the recipient of numerous honors and awards. Dr. El-Deiry has also trained many students and postdoc fellows, physician scientists, and continues to mentor junior scientists and faculty in basic and translational cancer research. There's so much more that I can say about Dr. El-Deiry and his research. I saw this as a major coup for Brown when we were able to convince him to come, and he has done such great things in the few years he's been here. So please join me in welcoming Dr. El-Deiry. Thank you.
Thank you, Rick. Can you hear me okay?
Yes.
It's great to be here at Brown, and I only wish we could be together in person today. What I thought I would do this afternoon is speak a little bit about cancer in general, give you some highlights of my research, and then share some developments within our cancer center here at Brown University. So let me okay. These are my disclosures, and I will give you a minute to look at them. So everyone's been touched by cancer. But what is cancer? Well, it's hundreds of diseases. Our cells transform, they pile up, they grow as a mass, they invade and metastasize. Cancers are like organs with a blood supply and many different cell types. If caught early, many cancers are cured with surgery. Metastases to other organs, for the most part, are what kill people and are hard to treat. Cancer is a disease of aging, but it can occur earlier in life when inherited or due to different exposures. Almost half of all cancers are preventable. Cancer is treatable, and some cancers are curable. Cancers evolve to more aggressive forms that become resistant to treatment. Research brings hope for ways of preventing and treating cancer. So these are just some statistics, the latest statistics from the American Cancer Society for 2021. So in 2021 in the United States, it's estimated there'll be about 1.9 million new cases of cancer and just over 600,000 deaths. And in the state of Rhode Island, we expect this year almost 7,000 individuals to be diagnosed with cancer and just over 2,000 deaths. This is the landscape of cancer incidence and mortality for men and women. So the most common cancers among men are prostate, lung, colon, and bladder cancer. For women, it's breast, lung, colon, and uterus cancer. Deaths in men are primarily lung cancer, prostate, colon, pancreas, as we go down the list. And for women, it's lung cancer, breast cancer, colon, pancreas cancer as we go down the list. Now, the survival depends on the stage of the cancer. So if a cancer is caught early, the 5-year survival is much better. If it's caught late with metastatic disease, survival is much, much worse. And you could see here 5-year survival. And again, these are the 2021 statistics: esophageal, liver cancer, lung cancer, pancreas, bladder cancer, cervical, uterus cancer, you know, is pretty much single digits. And I would say despite all of the progress that's been made over decades to diagnose and treat cancer. On a more positive note, lives have been saved because of research and advances. And again, this is from the American Cancer Society. Between men and women, almost 3 million lives have been saved over the past 3 decades due to research advances that have basically been flattening the mortality curves. This is the time of COVID, and that's brought some new challenges with reduced access to care, delays in routine care, later diagnosis, and some delays in treatment. The director of the National Cancer Institute, Dr. Sharples, has provided estimates of the impact of COVID that includes thousands more deaths over the coming decade, for example, here from colorectal or breast cancer. And it's noteworthy that people of color are disproportionately affected as far as these trends. Now, what I said earlier, that half of all cancers are preventable, the American Cancer Society has outlined the significant contribution of smoking and excess body weight, to both the incidence and mortality of various tumor types, as you can see here. Now, all cancers have consistent hallmarks that have been outlined by Hanahan and Weinberg over the last couple of decades. And these include changes within cells and tumors that include things like immortality, sustained proliferation of cells, genomic instability, inflammation, altered metabolism, and cell death resistance. And cell death resistance happens within the cells themselves, as well as to block the effects of the immune system. There are also hallmarks of metastasis that have been delineated by Dr. Welsh and his colleagues. These are features that tumors acquire, such as motility and invasion, modulation of the tumor microenvironment, plasticity, which basically means the cells change their form to help them get up and get into the circulation and end up somewhere else, and to be able to colonize. Now, cell survival is a hallmark of cancer metastasis because it contributes to the ability of the cells to metastasize. But in addition to the biology of cancer, there are also physical traits of cancer that have been described and summarized by Rakesh Jain and his colleagues last year in Science, including things like solid stress, fluid pressure, stiffness, microarchitecture, and evasion of cell death contributes to tumor progression and therapy resistance. So while cancer has existed since ancient times, we've learned much in recent decades. And the story or the mechanism really begins with changes in the DNA. So in the 1980s, cancer was understood as a disease of cell proliferation due to gene mutations. Chemotherapy killed dividing cancer cells. I studied mutagenesis in graduate school to learn about how mutations occur. And so mutations can occur spontaneously through exposure to environmental mutagens or as part of inherited or acquired characteristics of cells. Now, by the late 80s, cancer was being viewed as a balance of proliferation and cell death. So less death within a cancer meant bigger tumors. And oncogenes and tumor suppressor genes were becoming recognized basically as the accelerator and the brakes within a cancer cell. And this is when I became involved as a cancer researcher in the early 90s and worked with Dr. Bert Vogelstein at Johns Hopkins. And his early work unraveled the genetics of colon cancer as a function of progressive clinical and histopathological stages with which specific genetic alterations and oncogenes and tumor suppressor genes, as well as epigenetic changes, contribute to the progression. And this was truly a story of collaboration between gastroenterologists, pathologists, geneticists, laboratory researchers. In this model, this P53 over here is a tumor suppressor gene that is mutated in this sequence of events. And the sequence of events has become known as the Vogelgram in honor of Dr. Vogelstein. So by 1989, P53 was recognized as a tumor suppressor gene, and this hit the late press. And so this gene was discovered to be mutated in virtually all human cancers. And so there was interest in understanding how does this gene contribute to suppressing cancer normally. And so we would learn that gene mutations can actually be inherited in this gene in a syndrome called Li-Fraumeni syndrome, where individuals in successive generations are affected by various different common tumor types at a young age, such as breast cancer, colon cancer, leukemia, brain tumors, sarcomas, and many other tumor types. Some of my early work while at Johns Hopkins contributed to our understanding of how P53 works. And it was found to be able to bind DNA, and I was involved in elucidating the precise sequence that P53 could bind to in the human genome, and the specific amino acids that touched the DNA were actually the hotspots for mutation in human cancer, which told us that this property of P53 is very important. But what does it bind to, and what does it really mean as far as cancer? And so we work to understand what P53 binds to and what genes it turns on. And we discovered that P53 induces a gene called P21, which at the time I called WAF1 for wild type activated P53 fragment number 1. Induction of this protein, which is a universal cell cycle inhibitor, essentially blocks the cell cycle engine, leading to growth arrest and DNA damage. And this is a cell cycle here where after damage, you really don't want to be replicating your DNA in S phase and dividing with mutated DNA. And so this growth arrest after DNA damage allows cells time to repair. And if there's too much damage and not enough repair, the cells will die. When I started my own lab at the University.
Wafik, we're having trouble hearing you.
Oh.
Now we can hear you.
Okay, sorry about that. So we wanted to understand more about how cells die, how do tumor suppressors like P53 cause cell death to keep cancer from developing, how do cancer cells evade being killed by chemo or the immune system, and what are cancer's vulnerabilities that would allow us to develop new treatments. I'll tell you what we've learned over the years, and this is our current understanding of cell death as summarized by myself and my colleague Benny Carnero last year in Nature Reviews Clinical Oncology. And it's complicated. This is a mammalian cell. There are things that happen in the nucleus that send out signals into the cell. Things happen in the mitochondria, at the cell membrane. The cell takes cues from its outside environment, including things that promote cell death, such as signals from immune cells or the immune cells themselves, or things that block cell death by altering that physical aspect of the tumor microenvironment. Now, cell death pathway was elucidated in worms by Bob Horvitz at MIT, and he won the Nobel Prize in 2002 for unraveling the gene in C. elegans and for recognizing the conservation of this core pathway in flies and in mammals. Now, our own research has focused on how cells die after chemotherapy and radiation, and how tumor suppressors such as p53, can lead to cell death. And so we discovered death receptor 5, which sits on the cell surface and responds to a protein called TRAIL, which is part of the host immune system. And TRAIL signals death of transformed and tumor cells to suppress cancer and its metastasis. Now, this definitely was a group effort by many students and researchers in the lab over many years, as you can see here. Now, this is a very brief video just to give you a sense of how TRAIL kills some lung cancer cells over a 4-hour period in the lab. So these cells are definitely dead, and I think you could clearly see that. But we've wanted to do more with our knowledge to help patients. And so we wanted to see if we could develop new drugs building on the knowledge of these pathways. And so our strategy has involved doing cell-based screening with molecular readouts, identifying molecules of interest that modulate the cellular pathways to suppress cancer, prioritizing our compounds, and moving them to clinical trials. So we screened for TRAIL-inducing compounds. So normally our host immune system makes TRAIL, and this can bind to cancer cells and kill them. And about 12 or 13 years ago, we actually wanted to screen for compounds that might get the body to just make more TRAIL. And so we discovered this drug ONC201, which is also known as TRAIL-inducing compound number 10, that kills cancer cells but not normal cells. And this was published in 2013 and had effects in mouse models in various tumor types like colon cancer, breast cancer, lung cancer, lymphoma, and brain tumors. And the compound moved to the clinic and has been looked at in a number of tumor types with numerous clinical trials that are ongoing. The drug appears to be most active in some aggressive brain tumors. I think this is exciting, and for me as a physician scientist, it really is a thrill to be living through seeing something like that, where a molecule from our research lab ends up in the clinic and can do something like this in this young girl who couldn't smile because of this middle of the brain tumor, and over several months was able to smile again. And in other tumor types shown here, you could see shrinkage of solid tumor metastases, or some other tumors that in the tissues that got smaller in different cancer types, such as endometrial or prostate cancer. Now, what's really exciting is what's happening in these patients with brain tumors, and these are gliomas, aggressive gliomas that have a specific mutation known as H3K27M. And at a recent review of the data that has been accumulated, about 30% of patients have tumor regression and objective responses, and about 55% have disease control with this drug in a disease where these recurrent brain tumors, really, the patients don't really have good options for therapy. And some of these responses have been durable, lasting 3 years in this patient, and other patients have had control of neurological symptoms. And if you look at the overall survival of patients with these recurrent gliomas, the prolongation of survival by ONC201 TIC10 looks favorable when you look at how these patients do in general. You know, these are months as far as median survival, and this is 1,000 patients with these midline gliomas, and this is what ONC201 is doing to that median survival. So while this is an exciting story, and I'm thrilled to be part of it and would love to do more of this type of research and translation here at Brown, there are challenges in treating cancer. You know, when a patient with advanced cancer comes to the clinic, for some cancer types that are preventable, you know, this wasn't prevented. It wasn't caught early. The tumor cells evolve. The tumors are heterogeneous with many genetic drivers. There are other cell types within that physical environment that make tumors difficult to treat. The tumors evade cell death. They evade the immune system. There are issues with delivering drugs to tumors. There are toxicities from the treatments. And in the modern era, we have financial toxicity with how expensive drugs are, and we have disparities in access to care as some of the challenges. In addition, it takes a long time to develop new drugs, and you know, this is taken from a review that was just published in Cancer Discovery about what clinical trials are going to look like in the future. And we hope in the future it's going to take fewer years, but you can sort of see how much you start with when drugs are discovered and how many of those initial drugs end up into a clinical trial of any kind, and then ultimately how many are approved and then end up being used to treat patients. Now, historically, there have been a number of examples of successes, and this is one of the best known examples that occurred over a 40-year span. And given that I just came from Philadelphia, I've got to tell you about the Philadelphia chromosome, which was recognized basically under the microscope by Knoll and Hungerford back in 1960. And, you know, they saw this chromosome in patients with chronic leukemia. And it wasn't until the early 70s that Janet Rowley in Chicago, at the University of Chicago, realized that what this was actually a translocation. So a part of chromosome 9 went to chromosome 22 to create this short Philadelphia chromosome that you see here and that you see here. And it wasn't until the early 80s that we recognized that this chromosomal translocation encodes a fusion of two genes to create a new protein in the cells called BCR-ABL. And then it took additional decade to find a small molecule that could specifically inhibit ABL within BCR-ABL, and this led to this some refer to as miracle drug, Gleevec, to treat chronic leukemia, and this drug was approved in 2001. Now, you know, this took 40 years. We hope in the future it doesn't take that long. And there are other examples of successes in lung cancer with Tarceva and many other second and third generation compounds, and Herceptin in breast cancer. So I want to show you just for a few seconds an image of a cancer cell, and this is a T cell coming to kill that cancer cell. It turns red because its membrane becomes leaky and its morphology changes as it's undergoing apoptosis, which is programmed cell death caused by the killing effects of the immune cells towards the tumor cells. And the ability of the immune system to be harnessed to treat cancer has been one of the most important breakthroughs in this century. We now routinely use immunotherapy to treat many tumor types in the clinic. And this led to the awarding of the Nobel Prize to Jim Allison and Tasuku Honjo in 2018 for studies that led to the clinical implementation, the early scientific discoveries of how tumor cells block the immune system and how novel therapies can block the molecules that block the immune system to unleash the effects of the immune cells to kill the tumor cells. And this is having impact in the clinic across many tumor types. I had the privilege of meeting Dr. Allison a couple of years ago when I was at MD Anderson as part of the NCI site visit for their cancer center renewal, and this was definitely a highlight of that visit. So with the remaining time, I'd like to share where we are with the development of the Cancer Center at Brown University. And this is our vision, to build a world-class cancer program that brings basic discoveries, clinical, translational, and population research to reduce the burden of cancer in Rhode Island and beyond. Our academic center has investigators at Brown University, the medical school, the School of Public Health, the hospitals in our different affiliated health systems, working throughout the academic center to address the problem of cancer. And our Cancer Center basically brings them together to work within programs and to translate discoveries into clinical trials, and that can also impact on the population at large. We're doing this building on our strengths that include outstanding clinical care, a foundation of existing research, and incredible talent and commitment by our faculty throughout the academic center. And we have strengths in all of these areas that I'm showing here, in various tumor types and various basic biological processes, and in clinical trials, as well as risk factors that impact on cancer development. Our community has unique needs in the state of Rhode Island, having to do with things like how do environmental factors impact our population? Are there unique genetic factors within the diverse population? And how can we increase screening and prevention among different groups within our community to improve their healthcare outcomes? So this is a summary of some of the developments within our Cancer Center over the last few years, including some notable clinical recruitments that I'll show in a minute. The development of the joint program in cancer biology. I was recruited toward the end of 2018. We established research programs as one sees at NCI designated cancer centers around the country. We established translational research groups. We became a member in a precision oncology alliance that basically allows our scientists and clinicians to access genomic data, treatment data, and clinical outcomes to develop new biomarkers and treatments for cancer. We have been working on improving the management and oversight and review of clinical research. The Cancer Center at Brown was approved by the university early last year. We became members of the Association of American Cancer Institutes that currently has 102 of the leading cancer centers in the United States and Canada. We convened an external advisory board late last year to discuss our plans for NCI designation, and we have been making progress in terms of community outreach and engagement, ongoing faculty recruitments, and plans to integrate across the academic center. So we have world-class leaders for our research programs in cancer biology, cancer therapeutics, and population science. Very well known to the Brown community, Dr. Sedevi and Zitkovich, Dr. Carnero and Safran, and Dr. Aluwalia and Sarkar. And our research programs that, as I said, build on our strengths, have certain themes that reflect upon those strengths, including in cancer biology, aging and inflammation, stem cells and signaling, DNA repair and environmental exposures. Within therapeutics, we're interested in new drugs and early phase clinical trials to really bring new advances at the earliest time to try and help patients. And within our population science program, we have a major strength in the area of tobacco and smoking cessation and other modifiable risk factors, and population genomics and informatics. Our meetings bring together scientists, researchers, and clinicians across different disciplines to think about the important questions for specific tumor types and what we might be able to do here that would be unique at our institution to be able to have the kind of impact that we would like to be having. So this shows some of the faculty that have been recruited over the last few years, including some senior faculty across basic science, clinical oncology, and population research. And these are some of the more junior faculty that have been recruited in the last few years. And our approach as we have our program meetings and translational groups is to think about how we're going to address cancer here at Brown University and in Rhode Island. And it really goes back to understanding the biology and how cancers grow and develop and metastasize and become resistant to various treatments. We want to find those vulnerabilities to be able to kill cancer cells and boost the immune system so that we can have better outcomes ultimately. And really, for patients, this in part means individualized therapy, understanding specific genetic alterations that are unique to a patient's tumor, tailoring the treatment for that patient and their tumor, and getting that treatment to the patients and making sure that the treatments are active and specific. And this is a challenge, but as I showed you earlier, there have been successes, and we hope to do more of this to more specifically treat subsets of human tumors based on their unique makeup and genetics. Over the last year, we've had some invited outside speakers, including Dr. Kalin from Dana-Farber, who won the Nobel Prize in 2019, Dr. Weinberg, who not only figured out how to transform human cells, but also has been working on how cells metastasize and has also worked to define those hallmarks of cancer, and several other colleagues from University of Michigan, Dr. Mirajver, who works on brain cancer metastases from breast cancer, and Dr. Koumenis, who works on stress, cellular stress, and how this contributes to growth and metastasis. Dr. Polyak, who is focused on finding new ways to treat breast cancer, and Dr. Levine, who is working to understand how leukemias evolve and how to treat them better. And we also started to give out some pilot awards to investigators, again, across the different programs. And these awards are intended to support innovative, impactful, translational, or basic research that's relevant to cancer, that involves collaboration. And you know, the recipients in 2020 included Dr. Cho, Dr. Dawson, Dr. Liang, Dr. Raufi, Dr. Sahin, Dr. Tapinos, Dr. Hina Khan, and Dr. Yajima. And we're looking forward to see their work progress and succeed as a result of our support. And I'll just touch upon a couple of things. Rhode Island has one of the highest incidence of bladder cancer, and this is an area of strength here, but also an area where we need more intensive efforts to understand environmental, genetic factors, and ways of treating or preventing bladder cancer within our community. We also have high rates of lung cancer, and we need to do better with screening in underserved communities. Radon is an issue here, and we hope to understand how exposure to radon can lead to lung cancer at a genetic level. Our efforts have global outreach with the work of Dr. Susan Koo-Uven, who is working on detection of cervical cancer throughout the world. And we also have a number of very strong areas of research, including in neuro-oncology, pediatric oncology, radiation oncology, thoracic oncology, GYN oncology, both at Lifespan and at Women and Infants Hospital. And we have interprogrammatic collaboration within our cancer center, and this is some exciting preclinical work that involves the development of a new type of immunotherapy, and some really elegant preclinical work done in the laboratories of Dr. Jack Elias and Chun Lee, with work that is trying to translate this to patients. And we have also been involved with an analog of that ONC201 compound that I mentioned earlier, ONC212, which looks promising in pancreatic cancer. And we are working through funding by the Warren Alpert Foundation to take this discovery to a human clinical trial over the next couple of years. I should mention that Dr. Martin Weinstock recently obtained a $34 million collaborative grant from the VA to evaluate whether a topical medication can prevent common skin cancer. And we hope to hear from him soon at one of our program meetings. And I would invite you to log in to hear some of about his work. There's other exciting work and a strength at Brown, including in computational science, work of Dr. Sohini Ramachandran and medical MD PhD student Sahar Shahmadar. Basically being able from germline DNA sequencing, in our normal cells to be able to predict a response to immunotherapy down the line, which is really a fascinating direction for that research and a strength here at Brown in computational science and bioinformatics and genomics. This is a little bit about that Precision Oncology Alliance I mentioned earlier that involves numerous NCI designated cancer centers and that we joined by May of 2019. I serve on the executive committee for this national collaborative consortium, and we are involved in different types of genomics research with clinical outcomes, and there are many opportunities for our faculty across the academic center to be involved in different projects that will shape the future of precision oncology. We've been making progress with community outreach and starting in 2020, and this is continuing. And we need to have sustained efforts in community outreach so that we can impact on our community, but also really understand what are the obstacles and challenges that members of our community are facing, and how can we help them have better access and better outcomes as compared to other groups. Our plans moving forward involve integration of research across the academic center, program building, collaboration, innovation, growth, and community outreach. And really, what does the future look like? Well, at the center of it is our patients and how we would want to be treated ourselves when we go to the hospital or want to prevent cancer from developing. And basically, this involves outstanding research, applying state-of-the-art technologies, having a readiness with clinical trials to offer to patients, and having a variety of support services and information to tailor our treatments to patients, to be able to detect cancer early, treat it, and ultimately we would like to be able to prevent it in the future. So thank you very much for your attention, and I'd be happy to take any questions.
Great. Thank you so much, Wafik, for a really terrific talk and fascinating work. Really, I mean, I knew some of it, but just sort of seeing the whole kind of panorama was really very impressive. So thank you for all the work that you're doing and sharing it with us. We have time, about 10 minutes or so for questions. And so if people please use the raised hand function, I can call on you.
There's a question here. How is the community, or maybe you want to call on David Figelman?
Yeah, I was looking at the raised hand function. In the Q&A, I see that we have a question from David Egelman, who asks, how is the community brought into the process? It seems that a Rhode Island small state advantage would be a community university partnership to develop prevention and early diagnosis programs. So why don't you answer that?
Yeah, this is absolutely correct. Community outreach continues to evolve in terms of strategies of how best to do that. And you know, we have known about the disparities, and the problem has been how to best address them. And it's something that needs to be ongoing and involve really different aspects of the community. And so within our cancer center, Don Dizon, who is a leader in medical oncology and women's cancer, has graciously been leading this effort with developing a leadership group that includes members of the community with an agenda. And it's definitely a work in progress. And one of the guiding principles is to keep it up and to have frequent interactions with the community in various different venues to really understand what the needs are, what the obstacles are, to hear from different aspects and different people across the community so that we can begin to address the challenges. And so, you know, I thank you for bringing up the question, and if feel free to reach out and make any suggestions, and, you know, we could always improve what we're doing in that regard.
Great. Any other questions? Let me see if there's more. People want to raise their hand. Well, until then, let me ask a question, Wafik, which is something, I mean, you gave this, you know, wonderful sort of overview of the different research projects and the incredible impact that the research has had. How did you get into this field? Of all the fields of medicine, how did you end up zeroing in on cancer biology?
Yeah, that's a really interesting question. So I went into medicine, you know, I never really thought about doing anything different. You know, my parents were doctors and lots of family members were doctors. Throughout my training, you know, I guess I decided to go into oncology in the late 80s. And at the time, you know, I was interested in research, and there were a couple of areas where one could do research at that time and hope to have some impact, including, you know, the HIV epidemic and cancer research. There seemed to be so much to do at the time, and I really saw a great opportunity to get involved with an incredible mentor that really shaped the rest of my career. I've always sort of enjoyed clinical medicine, and I went into oncology also because you treat the whole patient as an oncologist. You know, you're not just focused on the kidney or the heart or you know, other organs. And I've always enjoyed that, and sort of that holistic approach that's incredibly rewarding. And I enjoy it till now, and I keep doing it to learn about what's going on, you know, on the ground, and how are we doing things? What are the current challenges? And there are so many.
That's great. Thank you. I see that Phyllis Dennery has a question. Phyllis.
Thank you, Wafik. I appreciated your talk. I am curious about your answer to this particular question. So some of these pathways that you describe are very global and very general, yet when we take the drug that suppresses one element of the pathway to the clinic, it has a very narrow and specific group of cancers that it is able to treat. Is there some insights there? And also, I want to add that in pediatrics, we use multi-pronged approaches for most of the things we do in a very protocolized way. Is there a way to integrate this into that model of a cocktail rather than a bullet, you know?
Yeah, thank you so much for asking that question. And you can rest assured that that's the question that we are asking every day in my lab in terms of what is special about these H3K27M mutated tumors. We've gained some insights into what's special about them. You know, they grow, most of them grow deep in the brain in a dopamine-rich environment. There's some data that the drug blocks a subtype of dopamine receptors. But your question is a very good one, because we discovered this drug. We screened for a drug that would potentially treat many tumor types. And now it's not that it does nothing for other tumor types. There is a signal in endometrial cancer and in prostate cancer. And, this is a big part of what my lab does, is try understand drug resistance and combination therapies. You know, this is what oncologists love to do, is to combine different drugs, and hopefully in the future those drugs are not so toxic. But, the cell death pathway is complicated, and there are many interactions, including the immune system. We did uncover an immune stimulation by the drug. I'd say we're still learning about how is the drug working, which is an amazing part of our drug discovery and development system in this country. You can show that a drug is safe and effective and maybe use it to help patients, and then still work on basic questions and figure out, how do we do things better? How can we get more activity out of the drug? We have thought of, for example, molecular mimics of that genetic alteration, which involves methylation and acetylation of that histone. So could we use histone deacetylase inhibitors or EZH2 inhibitors to mimic that defect to get activity in other tumor types? And, some of the work being done in the lab by pediatric hem-onc fellow Robin Borsak and pediatric faculty member in hem-onc Wenny Chang is looking at this in brain tumors and in sarcomas, hoping to develop new treatments for difficult to treat tumors that could be part of the path forward with new clinical trials. So it's very much a work in progress. And then, of course, we learn from the human clinical trials themselves, from the patient specimens, and the big question of why do patients respond or not respond? And that's definitely ongoing and a work in progress. And as I said, a potent analog appears to have activity in pancreatic cancer. We'll see how it works when it gets to patients.
Great. Well, thank you so much, Wafik, for presenting today. I think all of us benefited tremendously from learning about your research, learning about the incredible impact that it's already had, and really the exciting cancer center that you're building up here at Brown and in Rhode Island. I'm sure that I speak for all of us on this call that we are really thankful to have you in our community. So thanks for all the great work. Thanks for doing this, and everyone else, thanks for attending, and please stay safe. Take care. Bye-bye.
Bye-bye.